The South African National Biodiversity Institute is known as an institute that leads and coordinates monitoring and reporting on the state of biodiversity in South Africa, and making such knowledge and information available to all stakeholders. As custodians of our biodiversity, SANBI, together with BBSA and all biobanking institutions, will be able to encourage safe and responsible use of natural resources.
SANBI Biobank is based at the National Zoological Gardens campus in Pretoria. Our collection is from close to 85,000 individual animals and storing approximately 190,000 various samples from room temperature to minus 196 degrees Celsius. The individuals represent 221 families from nearly 750 genera with almost 1,000 species represented. These species include mammals, birds, reptiles, amphibians, and more recently marine and freshwater invertebrates. The sample reception laboratory is considered the only dirty area of the bank and is where all the biomaterials are processed for moving into the various banks. The freeze-drying lab lyophilizes samples for future resuspension and extraction and is just one of the ways samples are stored. The room temperature tissue bank stores various biomaterials in a controlled environment. These include hair and feathers, samples collected and curated for the Google Barcode of Wildlife project, blood on FTA paper, as well as the biggest veterinary histopathology formalin-fixed paraffin-embedded wax block collection. We have two DNA banks. DNA Bank 2 are for samples currently being sorted and catalogued, with provision made for fridge, freezer, or ultra-low temperature storage. The herpetology collection previously banked at Kirstenbosch is now curated at the Biobank, and this includes 17,500 samples of reptiles and amphibia. DNA Bank 1 is where samples are stored at ultra-low temperatures ranging from minus 80 to minus 150 degrees Celsius. Fibroblast samples are stored in a dormant live state and include the extinct western black rhino, 6 northern white rhino cell lines, as well as many other species. The dispatch lab is where we prepare sampling kits, as well as some of the media prep. The cryobank consists of 18,000 plus semen and biopsy straws submerged under liquid nitrogen, which is topped up weekly to maintain the collection. At these cryo temperatures, these samples are considered alive. Currently, the walk-in fridge houses our ethanol samples, and the walk-in freezer or the frozen post-mortem samples. And lastly, the team: Portia Choma, Katune Shunzi, Kenilwe Musupi, and Gaugelo Matloa, and I'm Kim Labashane. Thank you.
Welcome to the IMBM, or the Institute for Microbial Biotechnology and Metagenomics, here at the University of the Western Cape. We are situated in the Life Sciences building, and we are part of the Department of Biotechnology. I hold the position as the IMBM's Research and Innovation Manager, but I'm also proud to manage the IMBM Microbial Biobank.
As microbiologists, we get really excited about the incredible diversity of bacteria and their viruses, and especially through genome-guided discovery, using them as solutions to a more sustainable future for humankind, because their natural functions can be harnessed to make products and to tackle hard problems that are created by the growing human population and climate change.
My role is to manage the dedicated biobank equipment. This includes a shaking platform on which we grow our bacteria. We have a shaking incubator for bacteria that require a certain temperature to grow, as well as 2 dedicated minus 80 degree freezers. With regards to the culture collection, my responsibilities are threefold. Firstly, to confirm the quality of the isolates. My second responsibility is to capture all this information as well as any other data and enter it into the biobank database. At the IMBM, we continually grow our collection. This brings us to my third aim, which is to introduce the new strains into the biobank.
There are several programs running concurrently in the Institute that, as part of their activities, isolate microorganisms from a variety of environments. These bacteria undergo testing to determine if they have desirable bioactivities, such as producing antimicrobial compounds or other compounds of interest. As part of the characterization, we initially sequence the genomes of those bacteria with interesting bioactivities with the goal of eventually sequencing all of IMDM's collection. The screening information, together with the sequence data, all go towards building a biobank rich with data about the strains that we curate.
At the IMBM, however, we will never be able to fully utilize the potential that this collection holds. Therefore, as the Research and Innovation Manager, I'm highly delighted about the many opportunities that the Microbial Biobank provides.
SANParks is a government conservation authority with a mandate to conserve, protect, control and manage national parks and their biodiversity. Wildlife services form part of Conservation Services Department in SANPark. The VWS Biobank in Kruger is in Mpumalanga Province in the Kruger National Park's Kukuza. This unique and biodiverse environment of free-roaming wildlife offers a perfect opportunity to collect valuable samples. The aim of VWS is reintroducing populations, enhance conservation status of species, generate revenue through wildlife sales, and coordinate research on wildlife diseases. The implementation of these aims provide an ideal situation to collect opportunistic samples. This opportunity of taking samples started about 23 years ago, and in this time it has developed and transformed to be what it is today. In Kukuza, we have 4 laboratories available for researchers to use, and for our own needs, Zintle Manda, a biobank technician employed on the BBSA program, currently working hard at allocating samples for researchers. Intimate Shongwe, a veterinary research intern, is busy sorting and organizing hair samples. Tebogo Manamela, a veterinary technologist of SANParks, is receiving and biobanking new samples. Years ago, we were lucky enough to have a donation by the SANParks Honorary Rangers program convert a necropsy hall into a modern, isolated biobank building. Liana Fraser investigating some archived samples from the biobank. SANParks manages a system of 20 functional national parks in 7 of the 9 provinces of South Africa, with a total area of over 4 million hectares. To provide the same veterinary service in the southern parts of South Africa, BWS is also situated in the Northern Cape province in Kimberley. Kimberley VWS interact with a variety of unique animals, and therefore in 2012, Kimberley VWS started their own biobank facility.
The Department of Agriculture, Land Reform and Rural Development at Roller Park Evaluation Center established the National Gene Bank in 1998. Samples of seed and vegetative species are mainly obtained from communities farming under traditional farming methods to produce their food, and in some instances using their indigenous agricultural knowledge and practicing their cultural beliefs. When material arrives at the gene bank, samples are processed to determine their health and quantities. Currently, there are about 6,300 samples of seed and vegetative species of crops, medicinal plants, and some wild plant species. The gene bank maintains some vegetative samples that cannot be stored as seed. These are prepared in the laboratory to store buds or shoots in tissue culture. A sterile environment is necessary to prevent contamination and loss of sample during storage. As explants grow during storage, regular subculturing is performed as required to keep them alive from dying out. The gene bank uses a walk-in storage facility for active collection to allow for access to provide material for utilization. Sweet potato sample stores in tissue culture are duplicated in the field gene bank. When placing processed samples in freezers for long-term storage, samples are packed in 4 small bags for extraction every 5 years attached to a bulk bag. A duplicate sample in the National Gene Bank is deposited with the SADC Plant Genetic Resources Center in Lusaka for safekeeping. Data generated for each accession is documented on paper and the SADC Documentation Information System developed by the SADC Plant Genetic Resources Center. Additionally, the National Gene Bank reports the samples it holds to the FAO Early Warning System.
The National Research Foundation, South African Institute for Aquatic Biodiversity, or NRF SIAB, is situated in Makanda, a small rural town in the Eastern Cape province of South Africa, and is an internationally recognized center for the study of aquatic biodiversity, considering that we are most commonly known for housing of coelacanth specimens. NRF SIAB is also the recognized leader in wet collection management and curation in South Africa, and the specimens held in the JLV Smith Collections Management Center reflect SIAB's aquatic research over the past 50 years and are dominated by the National Fish Collection. Together with the wet collection, the NRF SIAB also houses a biobank facility. Samples of muscle tissue or fin clips from fish, frogs, and invertebrate species are stored in 99.9% ethanol in 2 ml Eppendorf tubes and placed into cryoboxes, which each hold a total of 81 tubes. In addition to these samples, we also store DNA extractions. Each sample is uniquely labeled with unique box locations. Related sample information is then captured on the tissue workbench spreadsheet, which is imported into the specified database and linked to catalog vouchers or observations. The NRF SIAB Biobank holds a total of 6-80 degrees Celsius upright freezers. Our first freezer was purchased over 10 years ago and can store a total of 40,824 samples. With funding provided from the BBSA, we were then afforded the opportunity to build on our current capacity and add a further 5 freezers to our fleet. Our freezers are constantly monitored using an online system connected to temperature sensors, and if temperatures drop below a predefined limit, we are sent email notifications so that the fault can be tended to immediately. Our biobank provides support to research projects by enabling access to our tissue samples through supplying gifts. Gifts can be provided internally to SIAB users or can be posted to other local or international institutions. The NRF SIAB Biobank looks forward to the continuation in building our relationships with the BBSA network to further enhance and broaden our functioning and relevance within a local and global context.